Hey everyone, this is Tiffany Watts. I'm going to be talking about case discussion number 2. The first case that I chose was case number 2 regarding the regulation of glucose uptake in the cell and in the glioblastoma tumors. So to start off, the uptake of glucose is regulated in liver, skeletal, muscle, brain, red blood cell, and adipose tissues, as we've learned in class. For the above tissues, glucose is regulated via glucose transporters, or GLUT. They act as facilitated glucose transporters by shipping glucose into a cell down its concentration gradient. Also, as we have learned in Unit 2 specifically, these tissues are fueled by dietary glucose with the specific mechanism of glucose uptake in each tissue. Just to quickly run over metabolism in the fed state regarding the mechanism of glucose uptake. So in liver tissues, they're going to be used by the GLUT2 transporters. In skeletal muscle and adipose, they're both going to use GLUT4, which are the insulin-sensitive transporters. Brain, they use GLUT1 and 3, and then in the RBCs, they use GLUT1. So since we're talking about the brain, let's talk about GLUT1. So GLUT1 expression reflects the level of glucose metabolism in a cell, is found in almost every tissue, and is highly expressed by erythrocytes, endothelial cells, and adult brain microvessels. They're encoded with a specific gene that functions as a uniport transporter, which these serve as important transport proteins in tissues, like the blood-brain barrier, which they can facilitate transport of D-glucose in the brain, and it's important to note that they're sodium independent. So just to quickly go over metabolism in the fasted state, so we talked about the fed state, and we're going to talk about the fasted state. So liver and skeletal muscles both use free fatty acids via lipolysis. Brain tissue and red blood cells also use the same pathway, so they use glucose via the hepatic gluconeogenesis and glyconeogenesis. Adipose tissues use free fatty acids via stored triglycerides, and here there's not as much energy required. So as we learned in week 4 of Unit 2, cells in normal tissues mostly produce ATP via oxidative phosphorylation in the mitochondria. And in this process, glucose is converted to pyruvate via glycolysis. Most of the pyruvate here enters the mitochondrial oxidative metabolism to generate efficient energy. This is where normal cells and cancer cells differ. So in the cancer cells, glucose is consumed through glycolysis, even if there is enough oxygen present, in the event referred to as the Warburg effect. The last step of this effect leads to the production of pyruvate and lactate as the final metabolites. So per research via Jin and Zhu in 2019, this enriched process of aerobic glycolysis creates an environment that gives cancer cells a better chance to multiply via generation of increased amounts of nutrient-rich materials such as ATP, amino acids, fatty acids, and nucleotides present. So the pentose phosphate pathway, which we've learned about in class, is a metabolic pathway like glycolysis that represents the first major step in the metabolism of glucose. It has an important role in supporting the growth and survival of cancer cells via generation of pentose phosphate, which is needed for the nucleic acid synthesis. So this pathway also provides NADPH or NAPDH needed for fatty acid synthesis and cell survival under stress conditions. So this flux of the pentose phosphate pathway is an important metabolic modification in several environmental frameworks and human malignancies, such as cancer, which is what this case refers to. So like we learned in Unit 2, the pentose phosphate pathway takes place in the cytosol, is made up of 2 distinct segments. It has an irreversible oxidative segment and a reversible non-oxidative segment. So these 2 segments are independent of each other. The irreversible oxidation portion converts glucose 6-phosphate to ribulose 5-phosphate via enzymes glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. During each reaction, the NADPH is produced via regulatory enzyme C6PDH. The production of NADPH inhibits the activity of that regulatory enzyme. The reversible non-oxidative portion converts ribulose 5-phosphate back to glucose intermediates, glyceraldehyde 3-phosphate, and fructose 6-phosphate, which we learned about in class. This is achieved via two primary enzymes, trans-ketoaldolase and trans-aldolase, and they have specific activities. So trans-ketoaldolase moves 2 carbon units, requires a primary enzyme. Requires use of a cofactor, is a measurement of erythrocyte activity, and measures the nutritional status of thiamine. Trans-aldolase moves 3 carbon units depending on its cellular need. The production of ribulose 5-phosphate is essential for amino acid synthesis. Just remember that moving forward. So normal cells have a lower rate of glycolysis that is ensued by oxidation of pyruvate in the mitochondria. Cancer cells, however, have a higher rate of glycolysis that is ensued by lactic acid fermentation in the mitochondria. In cancer cells, there's an overproduction, or kind of think of it as impersonating, of glycolytic enzymes. This impersonation of enzymes such as hexokinase 2 will work to increase the glycolytic rate and thus provide more energy into these cancer cells. So research performed by the Cancer Biology and Therapy Center proposed the idea that the high glucose uptake in cancer cells favors the cell proliferation more than energy production, because glucose metabolism mainly supports fatty acid and ribose 5-phosphate synthesis. Subsequent research noted that the increase in the glycolytic flux of cancer cells is partially caused by activation of regulatory enzyme fructose 2,6 biphosphatase. Fructose 2,6 biphosphatase is synthesized by the PFKFB enzyme and is an allosteric activator of the rate-limiting enzyme 6-phosphofructo-1 kinase, or PFK1. PFK1 is an essential point of control in the glycolytic pathway. This is due in part to the activity being inhibited by ATP. So PFKFB can halt inhibition, which in turn can boost glucose uptake and glycolytic flux. The metabolic reprogramming of tumor cells shifts the flux of energy, enhances glycolysis, and ultimately restructures the Krebs cycle due to the decrease in oxygen availability. So, another research found that the activation of hypoxia-inducible factor, or HIF1, in aerobic glycolysis regulates a response that involves the distorted expression of numerous amount of genes involved in glycolysis, such as glucose transporters, GLUT1 and 3, as well as glycolytic enzymes. So the lack of oxygen, or hypoxia, has also been found to induce pyruvate dehydrogenase kinases and inactivation of pyruvate dehydrogenases needed to convert pyruvate to acetyl-CoA. The severity and metastases of certain cancers has been linked to the amount of overexpression of HIF1. Some researchers found that upon culturing these tumor cells under hypoxic conditions, increased expression of GLUT1 was found. Thus, why we see a large area of white matter on MRI and large area of contrast on PET scans after the administration of labeled D-glucose. So that's case 2. Case 4, we're going to be talking about the jittery spells. So after reviewing the information taught in Unit 2 paired with research articles, I believe the child is suffering from glycogen storage disease. So the 2-month-old female presents to the ED with her mother with symptoms including no PO other than breast milk, change in sleep pattern, and labs consistent with hypoglycemia. While in the ED, epinephrine was administered with no increase in blood glucose, but it shows an increase in blood lactate. Workup shows that blood glucose increased after breastfeeding, but she was unable to maintain this blood glucose within normal limits during fasting. On physical exam, the liver edge was palpated 4 centimeters below the right costal margin, a physical exam finding consistent with hepatomegaly. So first I'm going to talk about the hereditary fructose intolerance and why I don't believe that's the answer. So patients with HFI have multiple symptoms including nausea, vomiting, diarrhea, abdominal pain, fatigue, often chronic malabsorption of nutrients. So breast milk contains a disaccharide carbohydrate made up of glucose and galactose. Due to the patient's symptoms consistent with this condition, an insignificant amount of fructose present in the breast milk, I do not believe that the child is suffering from this condition. Glucose 6-phosphate dehydrogenase deficiency patients often present with symptoms of fatigue and jaundice, and due to the association with red blood cells. So per the AAFP, lack of G6PD leads to hemolysis or destruction of red blood cells. The destruction of these red blood cells is often triggered by infections, foods, medications such as aspirin or NSAIDs. If the child, in this case, was suffering from this deficiency, one would expect to see more comorbidities such as possibly sickle cell anemia or just anemia. The Cleveland Clinic defines glycogen storage disease as a genetic condition where the body is not able to store or break down glycogen properly due to an enzymatic abnormality. GSD can affect multiple tissues in the body as there are 13 different types of this disease. In Unit 2, we learned that glycogen is stored in the liver and muscle cells and is broken down into usable energy, glycogen, when needed. The breakdown into usable energy is achieved via glycogen metabolism. So under aerobic conditions, pyruvate is converted to acetyl-CoA, which enters the citric acid cycle or is used for fatty acid. Under anaerobic conditions, pyruvate is converted to lactate. It's important to note that lactate is an important alternative fuel during hypoglycemic episodes. Hormones such as insulin, glucagon, cortisol, and epinephrine work to synchronize the relationship of glycolysis, gluconeogenesis, and glycogen synthesis. The administration of epinephrine increasing lactate is a result of the body trying to compensate for the inability to release glucose. Epinephrine is a hormone that stimulates the release of lactate from skeletal muscles needed for gluconeogenesis. The increase in lactate is a direct result of aerobic glycolysis, as lactate is an alternative during hypoglycemic episodes as mentioned above. So that's my synopsis of cases 2 and 4 for case connection number 2. Thank you.